like to introduce our next speaker, Dr. Devon Lawson. Dr. Lawson is an associate professor in the Department of Physiology and Biophysics within the Chow Family Comprehensive Cancer Center. Dr. Lawson has affiliations with the Center for Complex Biological Systems, the Stem Cell Research Center, and the Immunology Institute. Her laboratory studies the role of cellular differentiation in breast cancer initiation and metastasis. Dr. Lawson's lab is unique in its capacity to perform both high-level computational and experimental studies, enabling us to generate new hypotheses. Lawson has an extensive background in cancer biology. She did her graduate training under the mentorship of Dr. Owen Witt, where she worked to define prostate stem cells and interrogate their role in prostate cancer initiation. I'll now turn it over to Dr. Lawson.
Great, thank you so much. Well, good afternoon, everyone. My name is Devon Lawson. As you probably have already noticed, my background and function here at UCI is actually quite different than Dr. Tewari. I am what you would call a very traditional cancer biologist. So I did my PhD work at UCLA, where I studied prostate cancer, and then afterwards went up to UCSF to focus on breast cancer research. And like other scientists like myself, we spend a lot of time in the lab. We're very much experimentalists. We spend a lot of time on computers, crunching data that we get out of the lab. And so we actually, doing this is really a special opportunity for us to be able to share some of our research with the public, which is not something that we do every day. We don't see patients and communicate with the public. So this is really a wonderful chance to talk to you all. And I hope that I can teach you a few things about breast cancer research and some new findings actually that we've made in the last few years since I've been at UCI that we're hopeful will impact breast cancer patients in the short and long term. Okay, let's see. All right. So today, as I mentioned, I'm going to tell you a little bit more about our work specifically studying metastasis. And we tend to use, we heavily rely on new technologies in genomics, which I'll tell you more about in a few minutes. So metastasis, what's metastasis? We talked a little bit about that just a few minutes ago. Unfortunately, this is the phase of cancer, any cancer, particularly breast cancer, where the cancer cells actually spread. So you can see here in this image, the cancer cells in the breast actually can spread to other organs in the body. And this is really the main cause of fatality for breast cancer patients. Cancer in the breast itself is pretty treatable. I'll show you a little bit more on that in a minute. But as soon as the cancer spreads, it becomes very, very difficult for us to treat. And this is where we really go into chronic treatment phases, just really trying to manage disease chronically. So cancers can spread from the breast in a variety of ways. Here you can see in the image on this side, these blood vessels. They are what carry the blood to oxygenate all the organs in your body. Cancer cells can also spread through the lymphatic vessels. These are a vessel system that essentially connect all the lymph nodes in your body, which you can kind of see here on this side. The lymph nodes are where your immune cells lie. And so this is how the immune cells travel around the body. Unfortunately, cancer cells, because they've changed their biology, they can actually move from the breast through the lymphatic system or the blood vessels. They use these as highways to spread to other parts of the body. Normally, cells in the breast do not have this capacity. They're supposed to stay there in the organ and do their job, which is to produce milk, during pregnancy and lactation, after pregnancy. But oftentimes through DNA mutation, these cells acquire capabilities they're not supposed to have, and they can spread to other organs in the body. And just as a side note, this is why in the clinic, we often biopsy lymph nodes, so the lymph nodes around the breast, to see whether cancer cells have already started to travel around the body. This is a really good indication to us in terms of diagnostics. We're just talking a lot about tests, right, to determine, to diagnose disease or find disease. The same in this case for metastasis. a lymph node biopsy helps us determine whether the cancer has already started to spread throughout the body. So as I said, metastasis to vital organs is the main cause of death for breast cancer patients. So you can see here in this schematic published in the SEER NIH publications a few years ago, that actually breast cancer patients who come into the clinic with only disease in their breast, so the disease hasn't spread around the body yet, these patients do really, really well. We're very good at treating them with surgery and radiation. And 5 years after their diagnosis, 95 to 98% of them are surviving. So these patients we've gotten very good at treating disease at this stage. Unfortunately, patients who are diagnosed with metastasis, so where the disease has spread to distant sites, the situation changes really dramatically. So after 5 years after their diagnosis, unfortunately, the statistics are more around 25% or so of these patients are still surviving. And what's an even grimmer fact is the average survival for these patients is only about 3 years after diagnosis. And some of these patients who start out with localized disease in the breast eventually develop metastasis. So we really have a long ways to go in terms of being able to treat metastatic patients better. And unfortunately, about 40,000 or so women die of breast cancer metastasis every year in the US alone. And as I mentioned, kind of on this side, you can see in this line graph here where we're plotting time. So 5 years after diagnosis out to 17 years after the diagnosis of breast cancer. The percent of patients that recur, and you can see that the line just goes up and up and up. It continues sort of steadily over time. So patients can actually recur with breast cancer 17 years after being diagnosed initially. And they often recur with metastatic disease. So we really have to, the goal of research in my laboratory is really trying to figure out why this recurrence continues to happen after such a long period of time. And then we are trying to understand how cancer cells spread, and are there better ways to treat the spreading or prevent the spreading in cancer patients. So why is metastasis so deadly? I don't know if that's it's probably not obvious, right? I mean, why do we treat primary breast cancer so well, and we have such a hard time treating metastatic breast cancer? So as I mentioned, in contrast to surgery, where we actually remove the breast tumor surgically, we cut it out from the breast. When we have systemic disease and the cancer cells have spread throughout the body, we can't surgically remove them anymore. And we have to rely on drugs to try and kill them pharmacologically. And this is very, very difficult. And the main reason why it's so difficult is a principle that we call heterogeneity. And I was actually just thinking, as I was thinking about this presentation earlier today, about whether this is a lay term or a science term. I've lost track of that. But essentially, it just means that the cells are different. There are a lot of different kinds of cells within cancers. So within the tumor in the breast, there are a lot of different kinds of cancer cells that have different properties. And we've just drawn this here schematically in all these different colors. The cancer cells can vary a lot genetically. They have different DNA mutations. They metabolize nutrients differently. They interact with immune cells differently. The tumor is not one entity. It is a bag of cells that all can function quite differently. And when you take this, so essentially the problem here when it comes to trying to use systemic therapy, so drugs to target and kill these cells, the problem that we run into is we can't kill all of them because they're all so different. We can kill off a certain percentage of them that the drug is effective against, but we're left with the cells who are resistant and are not affected by the drug. So when it comes to metastasis and metastatic disease, we're even in like a whole nother realm of this problem of heterogeneity. So we know that individual tumors in the breast have this heterogeneity within them. But when we're talking about cancers that have spread throughout the body to different organs, this is compounded exponentially. So each tumor in the body then can have a bag of different cells with different properties. And so when we try and go in and treat patients with individual or multiple drugs, the chances that we can actually target and kill off all the cells in all these different places in the body becomes even more challenging. So it's really this problem of heterogeneity that we're faced with clinically, trying to treat patients with metastasis. And so in my laboratory, we've been using these new genomics technologies to try and study heterogeneity and understand the breadth of what kinds of cells there are in the system. So that if we can understand what kinds of cells are there, we think that we will be able to come up with better ways to kill all of them. Essentially, that's just kind of in a nutshell, the goal that we're going after. And a little bit about the genomics technologies that I find really amazing. We're in a really particularly exciting time in science with the explosion of new genomics technologies. There's really been a revolution in this field in the last 10 years or so, where we can do genome sequencing on a scale that we could never do before. So just for a little bit of perspective, I really like this because I grew up and graduated from college around the time that the Human Genome Project, the first human genome, was being sequenced and published. This took the field about 13 years. So they started it in the 90s, trying to sequence one genome from one person. It took 13 years and hundreds of labs worldwide to sequence this one genome, and it costs almost $3 billion. Today, we can do almost the same thing in my lab for like $100 in a couple of days, and we can do it from one cell. So it's really a crazy, amazing time in genomics that we can apply these new tools, this vast amount of sequencing technology, to understand all these different kinds of cancer cells and what they're doing and why they're different and what their therapeutic vulnerabilities might be. And just to give you another sense of all this is just a list of a bunch of these new genomics technologies that we have available to us that we've been using in my laboratory. So ranging from these microfluidics tools, where we actually capture individual cells in these tiny little micro wells, and we do the genome sequencing in these little tiny wells, to technologies where we capture individual cells in little water droplets. And we actually use the water droplets as little reaction chambers where we can do the sequencing of individual cells in these little water droplets. And even more recently, rather than having to take cells out of the breast and sequence them, we actually are starting to be able to sequence cells in place. So actually in situ on microscope slides or tissues that have been excised from patients, which gives us a whole nother level of resolution of heterogeneity of tumor cells. So in these technologies, we were looking at cells once we'd extracted them. We don't know where they came from exactly in the breast, but we know what they look like. With the newer technologies, we actually preserve spatial information, so we know how the cells relate to each other in space. It's giving us a whole new context of ways in theory that we can target these cells and kill them with drugs. So just to tell you a little bit more about a particular study that we published in my lab using these technologies a couple of years ago. We've been using this approach where we actually make what we call patient avatars. So we take patient tumor samples from the breast or metastatic sites, and we transplant them into the breast of host animals. And just to ease your mind about this, obviously I'm an animal lover myself. I have a dog, and we have a lot of pets at home. We treat the animals very well. They're very well maintained. We have a whole veterinary staff to monitor these animals every day. So the animals actually do very well here in our laboratory. But we use them essentially to make avatars of breast cancer. And what happens is the cancer cells that we transplant into the breast of these mice will actually spread to the same organs in the mouse that we observe in the patient that they came from. So this gives us a special opportunity to study metastasis in the mouse context outside of the patient and understand how cells in general, how they spread and what kinds of cells spread to different organs using these new genomics technologies that I mentioned. And here's just a picture of a little cluster of metastatic cells. They're a little bit different than the cells around them. These are lung cells here. And these are the little cancer cells that have spread to the lungs of these animals. And what we do is we use these genomics technologies to understand what kinds of cells are here and how they are different than other tumor cells in the body. So the first paper that we published a few years ago using this technology was actually done by one of my first graduate students, Ryan Davis, who's now actually a scientist in the Bay Area at a pharmaceutical company. And what he found using these technologies is that cells throughout the body actually have different metabolic properties. And particularly cells, when they're first trying to metastasize, they change the way they metabolize nutrients. And rather than using sugar as a nutrient source, which is very common for cells that want to grow, they adopt a different form of metabolism where they actually use the mitochondria to respire and create lots and lots of energy. So rather than eating a lot of sugar and creating byproducts from sugar metabolism, they use mitochondrial metabolism to make a lot of energy. And this energy essentially allows them to metastasize. It's like a fundamental property that enables them to spread. They need this extra energy. And so after we published this paper, we've been looking to see whether we can actually target this property of cancer cells, that they need to use this type of metabolism in order to make energy. If we target this type of metabolism and prevent them from being able to do that, can they no longer metastasize? And I have a graduate student and a postdoc that have been working on this project. It actually was itself a pilot grant from the Anti-Cancer Challenge in 2021, where we were looking to see whether these drugs that block mitochondrial metabolism can prevent metastasis in the animal avatars of human disease. And our initial experiments are very promising, so we're continuing this work now. You can just see in this little bar graph here that the animals that have been treated actually develop very, very little metastasis in contrast or comparison to the animals that were not treated with the drug. And we published this initial finding in 2022 in Communications Biology. So that's really the first step of this project that we've been working on. And so in the last couple minutes, I'm going to tell you a little bit about a different project where we are using the same technologies to study a different type of metastasis called brain metastasis. And this type of cancer, this type of metastasis, unfortunately, is very, very, very deadly in cancer patients. So about 30% or so of our patients develop brain metastasis. That translates into about 150,000 new cases of brain metastasis every year. So it's a big clinical problem. Most conventional therapies actually don't cross the blood-brain barrier, so they can't even get into the brain in order to treat and kill the cancer cells. So patients with this type of metastasis have a very, very, very rough time, and median survival is only about 5 months. So we've been working really hard to apply these really exciting new technologies to understand what's going on in brain metastasis in these patients. And just to give you a sense, this is an ongoing and newer study in my lab. We found that the cells that go into the brain, the cancer cells, they interact with this type of immune cell called microglia. It's a special immune cell that resides only in the brain. You can see the cancer cells here in green and the microglia surrounding them in red. You can see as the cancer cells grow out in the brain and they expand, you see more red microglia surrounding them. Previous work suggested that microglia are actually promoting disease, but what we found using the single cell genomics tools is quite the opposite, that these immune cells are actually surrounding the cancer cells because they're trying to kill them. They're trying to flag the immune system to target and eradicate the cancer cells. And so in the last minutes, I'm just going to basically jump to the chase, which is that we're using this finding now to see if we can target microglia and their ability to flag these cancer cells in order to improve immunity against brain metastasis. And we're hoping that we can start to apply immunotherapies better in the brain metastasis setting to make more of an impact and treat these patients better. And I think with that, I'm going to go ahead and thank everyone involved in the project. So this is the funky bunch of scientists that have been involved on these various projects. And so you can see us here, actually, I think this is from 2017 or 2018 Anti-Cancer Challenge, where we had our little pink capes and hats, and we tried our best to run and keep up with the flock. Here was a picture of a lab meeting that we did. We did 2 years of Zoom lab meetings during the pandemic. And this was an image from one of them where we were very enthusiastic initially, and we had little dress up competitions that waned, as you can probably imagine, after the first few months of the pandemic. And then, of course, I'd like to thank all of our funding, particularly the Anti-Cancer Challenge, which, as Dr. Tawari mentioned, has really been critical to provide funding for these pilot projects, like I just showed you, to test new ideas and new questions that we don't have federal funding or other funding sources to go after. And I think that's it for my talk. I'll be happy to take any questions if anyone has any questions on the research or anything in the area.
Well, thank you so much, Dr. Lawson, for your fascinating presentation. We do have several questions. So the first one is, how many different types of breast cancer are there?
Yeah, that's a great question. So I talked a lot about how different the cells are within a tumor, but actually even across patients, breast cancers are quite different. And there are 4 well accepted types of breast cancer, 3 to 4: basal-like breast cancer, luminal-like breast cancer, and HER2 positive breast cancer. And once you receive a diagnosis for the subtype of breast cancer that you have, we then have different kinds of drugs that we know have efficacy against the different types. And so there are about 3 or 4 subtypes. That being said, actually using these genomics technologies that I just mentioned, we're starting to see a level of resolution much beyond that. So like there was a paper that just came out a couple of years ago where they suspect there's actually something like 10 subtypes of breast cancer. Mm-hmm. And that as we start to be able to bin people, precision medicine better, we're going to be able to treat them with more effective drugs. That's a really good question.
Well, thank you. So the next question, should all breast cancer patients have genomic testing?
That's a really good question. In our clinic, we're starting to do this more routinely. A clinician could speak to that better. I think that as we move forward, absolutely, we will absolutely be doing this on all patients for a number of reasons. Right now, we have a certain number of drugs which we know we can treat patients with. And so the genomic information gives us all this information, but we don't have more drugs to work with, right? So we don't necessarily at this point in time benefit a lot more with the genomic analysis. However, as you can see from those charts, breast cancer patients live 20 years, 30 years, they live sometimes, forever. And so moving forward, having that genetic information is gonna benefit patients, I think, because like we're gonna come up with more and more ways to treat patients. And so the genomic information over time historically is gonna benefit us a lot, I think, moving forward.
Thank you.
Yeah.
Is the BRCA gene Dr. Tewari talked about for ovarian cancer the same BRCA gene for breast cancer?
Yes, absolutely. Yes, there is a particular type of breast cancer which is hereditary. It's inherited, and it's caused by specific mutations in the BRCA1 gene, and that gene also, if you have those kinds of mutations, also predisposed to ovarian cancer as well. So exactly, that's exactly correct.
Thank you. And then you did touch on this a little bit earlier, but are these research projects that you discussed today funded by the government or with private funding?
Great question. So both. The funding model, I think, as maybe actually Dr. Tewari talked about even better than I did, really is that when you have a new idea, for example, that came out of our sequencing study, right? We had this idea that we could maybe target metabolism and make an improvement in therapy, right, against metastasis. We didn't have funding to actually test those drugs, right, and see whether they could block metastasis. And we got funding through the Anti-Cancer Challenge and other private donors as well to start to do some of those experiments. And then what happens is those initial experiments that look promising enable us to apply for larger federal funding. And so it's really, that's been a great model for pushing research forward, starting with, philanthropic donations to really make those first exciting discoveries, and then request larger funding amounts to do the bigger studies afterwards.
Wonderful, thank you. And then last question, can you share, this might be looking to the future, but any other new breast cancer studies that you're working on your lab now?
Sure. Well, the brain metastasis study is one, obviously, that we haven't published it yet. We're getting closer. There's been a lot of new things that have come out of that we're interested in. So I talked about microglia, for example. They're an immune cell in the brain that we think we might be able to harness to prevent brain metastasis. But through those studies, we've actually learned about a couple of other immune cell types that go into the brain. Natural killer cells is a big one, and regulatory T cells. And so we actually started 2 new projects looking at those kinds of immune cells and to see whether we can actually target their capacity to kill cancer cells or alter the immune microenvironment. So we have a couple of new projects on other types of immune cells in the brain to see whether we can make more progress, basically targeting them as well. And we have other projects ongoing. Those are kind of some of the main, I think the main ones. So good question.
Thank you. And thank you so much for your time today and your continued support of the Anti-Cancer Challenge.
Great. Well, thank you all so much for coming out.
Thank you. And we're very excited to announce that the 7th annual UCI Anti-Cancer Challenge will take place on Saturday, October 7th in UCI's Aldrich Park. It will be a hybrid event with opportunities to participate both in person and virtually. In-person challenge day will include a 5K, 10K run walk, 14, 35, 60, 100-mile bike routes, a new mountain bike route, which we're excited about, entertainment, family-friendly festival, awards, and more. Next slide. So we hope you'll consider starting, joining, or supporting a team, and all proceeds support promising pilot studies and early phase clinical trials that can help prevent, treat, and cure cancer. Next slide. So we are hosting 3 more educational webinars featuring our renowned faculty speaking on hot topics in health, so more information can be found on our website. Next slide. And then we're also hosting several free in-person events leading up to Challenge Day, including rallies where you can participate in a training ride, run, or walk, and a research reception where you can tour the cancer research labs on UCI's campus. Next slide. So thanks again very much for joining us, and please scan the QR code listed here to receive more information. Have a wonderful day.